Imagine you're just, staring into an aquarium, and it's filled with this school of fish.
Okay.
Their primary pattern is this really stark, high-contrast black and white.
Right. Very striking.
Exactly. But suddenly, one of them turns, and you catch this flash of a bright, neon orangish hue right on its tail.
Oh, yeah. That always catches the eye.
It really does. Your brain just instantly locks onto it. You immediately think, well, that's a signal. That has to mean something, right?
Naturally. I mean, we're totally conditioned to do that.
We really are. In biology, we just assume that the loudest visual signal in the room is the answer to our question. But what if I told you that sometimes that glaring, brightly colored signal is actually a total biological lie?
Oh, man. It is a phenomenal trap.
It really is.
We are so wired to look for the most vibrant heuristic like that quick visual shortcut that we completely bypass the subtle structural realities that actually define what we're looking at.
Which perfectly sets up what we're doing today. Welcome to today's deep dive, everyone. Today we are exploring this fascinating entry from a personal journal. It was published on June 25, 2026 by a writer named Ang.
Right.
And the publication is called Notes on Fishkeeping. Specifically, we are looking at part 279 of this journal.
Which is just an incredible amount of dedication, by the way.
Oh, totally. 279 entries. And the mission of this specific entry, well, it sounds incredibly niche at first glance. Ang is trying to explain how to properly identify the gender of the polar parrot cichlid.
Yeah, which is a surprisingly complex task. And Ang is clearly a bit frustrated by how often people get it wrong.
Yeah, definitely.
But the underlying mechanics of why people get it wrong, I mean, that is what makes this source material so compelling. We're dealing with observer bias, the phenotypic expression of hybrid genetics, and even the rigorous physics required to actually document reality accurately.
It really escalates quickly from just looking at a fish. So let's jump right into the trap itself. Ang explicitly points out that a massive chunk of the fish-keeping community is getting this wrong.
Oh, yeah. Completely wrong.
They are looking at these polar parrot cichlids, trying to separate the males from the females, and they are relying heavily on that orangish hue on the tail.
Right.
Ang actually writes, as we've learned, coloration, or specifically an orangish hue on the tail, is not a good indicator for gender identification.
And that's such a tough pill for people to swallow, because the visual processing center of the human brain, it just loves a color code.
Oh, for sure. It's like assuming a car with a flashy racing stripe is automatically the fastest one on the lot.
Exactly.
When really it's just paint. So let's unpack this a bit. If this orangish hue doesn't tell us the gender, why are our brains so hopelessly wired to look for the flashy color first?
Well, think about cognitive load. When we look at a complex dynamic system, like a dozen fish darting around in water, our brains get overwhelmed.
Right. It's a lot of data to process all at once.
Right.
Exactly. So we desperately want a simple categorization tool. If we see a splash of distinct color on an otherwise monochrome organism, we just project functional meaning onto it. We assume, nature naturally color-coded the males or the females just for our convenience.
I understand the instinct, I really do. But if Ang is having to write about this in part 279 of a journal, people are clearly stubbornly clinging to this orange tail theory.
Oh, they don't want to let it go.
Why is it so hard for people to drop a visual clue even when an expert outright tells them it's wrong?
Because discarding a high-contrast visual clue means you actually have to do the work.
Ah.
Right. If color isn't the deciding factor, you are forced to look at geometry, structural details, and relative proportion. And that requires a sustained attention span.
Which nobody wants to do.
Right. It is much easier to just point at the orange spot and feel confident, even if your confidence is entirely misplaced.
It totally reminds me of looking at a complex data set. Like, say you have a massive spreadsheet or a heat map.
Oh, yes.
If there's one cell highlighted in bright red, your eyes just snap to it, and you assume it's the most critical piece of data.
Naturally.
But what if it's literally just a formatting error? What if that red cell is completely irrelevant to the actual trend you are supposed to be analyzing?
You've just let a superficial visual marker derail your entire analysis.
Exactly.
And honestly, that happens in data analysis constantly, and it absolutely happens in biological observation too. Ang is basically begging the reader to ignore the glaring formatting error on this fish's tail.
Okay, but wait, here's where it gets really interesting, and I have to push back on this idea of it being an error.
Okay, sure.
Because evolution is usually, ruthlessly efficient, nature doesn't typically waste metabolic energy generating bright pigments just for the fun of it.
That's a very fair point.
So if this orange tail is a false flag, why is it even there? Are you saying nature just leaves useless code running for generations without any purpose at all?
So in a pure, isolated evolutionary line, you would be absolutely right to be skeptical. But Eng provides a really critical piece of context here.
Okay.
The Polar Parrot Cichlid is a hybrid.
Ah,
It is not a naturally evolved species in the traditional sense. It is a crossbreed.
yeah.
And hybridization completely rewrites the rules of phenotypic expression.
Okay, unpack phenotypic expression for me in this specific context.
Mm.
How does being a hybrid sort of excuse the biological lie of the orange tail?
Well, when you cross two distinct genetic lineages to create a hybrid, the resulting organism inherits this massive, conflicting library of genetic instructions.
Like a mashup.
Exactly. The primary instruction that won out for the Polar Parrot Cichlid is that black and white pattern. That is its dominant visual characteristic. But the genes from its ancestors, they don't just vanish.
Right.
They are still sitting right there in the genome.
So the orange is coming from an ancestor.
Yes. In whatever ancestral species originally carried that orange pigment, it likely had a very specific crucial function. It might have been a mating display tied directly to gender, or maybe a territorial marker. But when those genes were folded into the hybrid mix of the polar parrot, they were decoupled from their original functional context.
Oh, wow.
So the genetic instruction to produce orange pigment on the tail is still randomly firing, but it is no longer tied to the sex chromosomes.
That is just fascinating. It's almost like a genetic ghost crashing the party.
Oh, I love that, a genetic ghost.
Right. The biological software is still executing an old line of code, rendering this random orange pixel, but the original hardware it was built for is completely gone.
That is a brilliant way to conceptualize it. The mechanism of inheritance in hybrids is rarely a perfectly smooth blending of traits. It is often a very messy living patchwork.
Yeah.
The alleles responsible for that orange coloration are expressing themselves randomly across the population, completely, regardless of whether the fish is actually male or female.
Which totally explains why observers get so hopelessly confused.
Exactly.
I mean, if you don't understand the underlying genetics of a hybrid, you look at that orange tail and apply the rules of a naturally evolved non-hybrid species. You just assume the color must have current utility.
You assume the organism is a finished, refined product rather than a genetic mashup carrying around obsolete historical artifacts. Eng is really forcing the observer to detach from the assumption of perfect biological utility.
Okay, so if the color code is a lie and the orange is basically just an ancestral ghost, I am now staring at a tank of 20 supposedly identical black and white fish.
Right.
How am I actually supposed to tell them apart? Aang eventually takes the training wheels off and tells us what we actually need to look for, right?
Yes, and this is where the observation shifts from passive looking to active structural analysis. Eng highlights two specific indicators, dorsal fin geometry and relative size.
Okay, let's start with the fins. Aang says we need to look for pointed or elongated dorsal fins.
I want to drill down into the why here.
Why the dorsal fin? Is there a biomechanical reason males might have a different fin shape, or is that just another random trait?
Well, while Eng doesn't dive too deep into the evolutionary biomechanics in this specific excerpt, in the broader context of cichlids, fin geometry is highly functional.
Really?
Yeah, the dorsal fin isn't just a stabilizer for swimming. During social interactions like territorial disputes or mating rituals, these fish will flare their fins in a lateral display to appear larger and more dominant.
Oh, so it's about posturing.
Exactly. A pointed, distinctly elongated dorsal fin alters the entire silhouette of the fish. It creates a sharper, more aggressive profile compared to a rounded fin.
So we are moving away from a superficial paint job, the color orange, to analyzing the actual structural engineering of the organism.
Precisely.
But I have to ask, how easy is that to actually see? Because fish don't exactly freeze in place and pose for you in profile.
No, they do not.
Judging whether a fin is slightly pointed or slightly rounded on a moving target sounds incredibly difficult.
It is incredibly difficult, which is precisely why Eng introduces the second necessary factor, which is the relative size of the fish within the population.
Okay.
One morphological trait on its own, especially in a hybrid where traits can be somewhat muddy and inconsistent, is rarely enough for a definitive identification.
I really struggle with this second metric, honestly, because relative size seems like a wildly unreliable variable to me.
How so?
Well, what if you just have a really well-fed female, or maybe a relatively young, stunted male? How do you index relative when you are dealing with a dynamic environment where everything is growing at different rates.
And that is the crux of the challenge. You cannot evaluate a single polar parrot cichlid in a vacuum.
Right.
If I hand you one fish in an isolated container, you have zero context for its size. You don't know its age, its diet, or its genetic potential. Eng stresses that you must look at the size relative to the rest of the population.
So it's like trying to guess if someone is tall without seeing anyone else in the room.
That's a great analogy. Yes.
So you have to establish a baseline. You basically need a cohort.
Precisely. You need a group of fish that have been raised in the exact same environment, subject to the same water parameters, and fed the exact same diet over the same period of time.
That makes sense.
Only then does size become a viable data point. You are performing a macro observation of the entire tank to establish the statistical norm, and then you are layering your micro observation, the fin geometry, on top of it.
Ah, I see. So you are looking for the intersection of two distinct data sets. A fish that is structurally larger than its identical age peers and possesses the elongated dorsal fin.
Exactly.
If those two vectors align, you finally have your answer.
And notice how much more cognitive effort that requires than simply spotting an orange spot.
Oh, it's a completely different level of observation. You have to synthesize population dynamics with microanatomy.
Right.
Which brings us to the documentation of all this, because understanding the traits in your own head is one thing, but Eng's entire goal is to teach others how to see this. And this is where the journal entry takes a really inspiring turn, I think, into the methodology of the scientific observer.
Eng is really grappling with the friction of knowledge transfer here.
Yeah, the entry begins with Eng saying, "Even though I've discussed this numerous times, I've finally taken the photo to showcase how to identify the gender."
You can almost feel the exhaustion in that sentence.
You really can. They have tried explaining the dorsal fins and the relative size in text over and over, but words alone just aren't cutting it. People are still falling for the orange tail, so Eng resorts to photographic evidence.
But then Eng immediately critiques their own photographic evidence.
Yes.
The very last sentence of the entry is a note to self for the future. They write, "For future posts, we should take more photos from a flat angle of different fish species, as this is easier for our discussion and learning."
That specific phrase, a flat angle, really caught my attention. I mean, we all know taking a picture of a moving fish is hard, but why is a flat angle specifically so critical for this type of learning? Is it just about getting a clear shot?
No, it is entirely about physics. Specifically, the physics of light refraction.
Okay, walk me through that.
Think about the medium you are trying to observe this fish through. You have a subject completely submerged in water, sitting behind a thick pane of glass or acrylic, and you are viewing it from the air outside the tank.
Right, so that's 3 different mediums with completely different densities.
Exactly. And light travels at different speeds through each of them. When light passes from water to glass to air at an oblique or skewed angle, it bends.
Oh.
It distorts the image. If you take a photograph of a fish from, say, a 45-degree downward angle, that optical distortion might stretch the image of a rounded dorsal fin, making it look elongated.
Wait, really? So the refraction could literally fabricate the very anatomical trait you were trying to use for identification?
It absolutely can. Furthermore, an angled shot completely destroys your ability to judge relative size because of forced perspective and refraction. A slightly smaller fish, situated near the front glass, might look structurally massive compared to a genuinely larger fish swimming an inch further back in the tank. The skewed angle corrupts your entire macro observation.
Wow. So a flat angle, like shooting perfectly perpendicular to the glass at a 0-degree angle to the normal, is the only way to minimize that light bending.
Yes.
It ensures that the geometry you capture on camera is the actual geometry of the organism. That is just an incredibly rigorous realization.
It is the realization of someone deeply committed to the integrity of their data. Eng recognizes that an observer's tool, the camera lens, can introduce a bias just as severe as the cognitive bias of the orange tail.
And what absolutely blows my mind is the context of when this realization is happening.
Right.
This is part 279 of Notes on Fishkeeping. Eng has written 278 previous entries. They possess a towering amount of domain expertise.
And yet here they are, still actively refining their methodology. They aren't resting on their laurels at all.
Not even a little bit. They are analyzing their own instructional materials and saying, the physics of my photography is flawed. Next time, I need a flatter angle.
It beautifully illustrates the difference between simply accumulating facts and truly mastering observation.
Yeah.
I mean, you can memorize the fact about the dorsal fins on day 1, but recognizing how environmental distortion, biological red herrings, and your own observer bias all conspire to hide the truth. That takes the sustained dedication of 279 journal entries.
It really is a masterclass disguised as a niche aquarium post. So let's trace the arc of what we've uncovered today from this single, concise excerpt. We started by dismantling a powerful visual heuristic. We learned why we absolutely must resist the urge to trust the loudest, most colorful signal in the room.
And we explored the messy, complicated reality of hybrid genetics, too. Understanding how an organism can manifest an ancestral phenotypic trait, a genetic ghost, that has absolutely no functional bearing on its present state.
From there, we mapped out the actual structural markers required for truth. We moved from the macro observation of establishing a relative baseline within a controlled population all the way down to the micro observation of analyzing specific anatomical geometry, like the pointed dorsal fin.
And finally, we explored the physics of documentation, revealing how water refraction and oblique camera angles can warp biological reality, making the pursuit of a flat angle essential for transferring accurate knowledge.
For you listening right now, the applications of this go far beyond identifying the gender of a polar parrot cichlid. Whether you are analyzing a volatile financial market, trying to debug a massive piece of software, or just navigating complex social dynamics at work, this deep dive is really a blueprint for critical thinking.
The brightest, most obvious piece of data is rarely the whole story. And honestly, it is often a complete distraction.
You have to look past the superficial paint job and study the structural engineering. You have to understand the context of the population before you judge the individual, and you have to constantly question whether the angle from which you are viewing the problem is distorting your perception of reality.
It takes immense patience.
It does, but it is the only way to separate the truth from the noise.
It requires a willingness to just sit in the ambiguity until the real patterns finally emerge.
As we wrap up today, I want to leave you with a thought about that concept of the genetic ghost. We've established that this hybrid cichlid is swimming around displaying a bright orange ancestral trait that serves no modern purpose. It's basically just an obsolete line of biological codes still running in the background.
Right.
It makes you wonder, what other obsolete genetic ghosts are hiding in plain sight, and not just in hybrid animals. Think about human biology or even human behavioral psychology.
Yeah.
What flashy, deeply ingrained traits or reactions are we carrying around every single day that have completely outlived their original evolutionary purpose?
We might all be responding to signals that haven't actually mattered for millennia.
It is a wild thing to consider. Keep questioning the obvious signals, keep searching for that flat angle, and we will catch you on the next deep dive.